The endoplasmic reticulum is the most extensive membrane system in the cell, and serves as an entryway for proteins being shipped to other organelles and for itself as well. Within the endomembrane system, the ER can ship proteins via transport vesicles to organelles including the Golgi, endosomes, and lysosomes. Messenger RNA exits the nucleus and attaches to a ribosome. A translation of a polypeptide chain commences. At the end of the growing polypeptide is a signal sequence which binds to a signal recognition particle, SRP, thereby pausing translation. The complex moves to the endoplasmic reticulum, RER, where the SRP binds to the SRP receptor on the RER membrane. The SRP receptor moves the complex along the membrane to a translocation
The
channel.
SRP and the SRP receptor detach. The translocation channel plug detaches from the translocon, allowing for translation to resume. The polypeptide chain lengthens the translocation channel into the lumen of the RER. The signal sequence is clipped from the polypeptide by the signal peptidase. The oligosaccharyl transferase transfers an oligosaccharide from the dolichols phosphate, the lipid carrier of the oligosaccharides, to the polypeptide. Glucose monomers hydrolyze off of the oligosaccharide, leaving one glucose monomer to bind the unfolded polypeptide to the calnaxium. The ER chaperone, which folds the polypeptide, also protein disulfide isomerase, forms disulfide bridges within the polypeptide, also contributing to the correct folding. The glucose cleaves off, allowing the protein to detach from the calnaxium. If the polypeptide is properly folded, a mannose monomer will cleave off, and a transport vesicle enclosing the folded polypeptide oligosaccharide complex buds off the RER membrane, which travels along the cytoskeleton to the Golgi apparatus, where the oligosaccharide undergoes transformation. The finalized oligosaccharide serves as a signal for the protein's final destination. The complex exits the Golgi in another transport vesicle to which Rab and V snare proteins are linked. The vesicle moves to the destination along the cytoskeleton. As the vesicle approaches the plasma membrane, tethering protein connects the Rab protein of the vesicle to the Rab protein of the plasma membrane. The V snare of the vesicle intertwines with the T snare of the plasma membrane, docking the vesicle. The vesicle fuses with the target membrane and the cargo exits the cell in exocytosis.
What you gonna do with all that junk?